Hi, I'm Frank Chiaramonte, and I'm the Program Executive for NASA's Physical Sciences Research Program, and we're part of the Space, Life, and Physical Sciences Research and Applications Division. We are interested in conducting biological and physical science experiments that will use the light microscopy module in a microgravity environment on the International Space Station. In order to facilitate this process, NASA is going to be releasing a research announcement soliciting proposals in two distinct areas, one complex fluids, the other macromolecular biophysics. In complex fluids, investigators will be able to submit proposals to participate in the experiment called the Advanced Colloids Experiment Test Series. In this experiment, we'll be observing the behavior of colloidal systems using a microscope in the microgravity environment of the ISS. By performing these experiments, specifically known as soft condensed matter experiments, scientists are able to remove the masking effects of sedimentation and convection and particle jamming in a timescale not available on Earth. The intent of the experiment is to observe the colloidal processes, to understand them, to learn how to control them. The ACE experiment will observe the behavior of a solid phase of microscopic particles dispersed in a liquid, which in microgravity remains suspended in the liquid for prolonged periods of time. Under these conditions, the data obtained will advance the understanding of rigidity, self-assembly, pattern formation, origins of dissipation, nucleation and growth, phase separation, and formation of crystalline structures currently not observed in Earth. The colloidal systems can be used to model atomic systems and make new structures using self-replication and self-assembly, leading to novel items such as nanopumps and photonic crystals that can be used for future optical computing. The second theme area is going to be macromolecular biophysics. In this area, scientists are invited to propose investigations of biological macromolecules, such as proteins, using the LMM on the space station. By removing or greatly reducing the effects of buoyancy-driven convection and sedimentation in microgravity, the results obtained will improve the understanding of the crystallization process and the diffraction quality of the resultant crystals. Research areas of particular interest include transport phenomena in protein solutions, protein aggregation, or how they come together, the fundamental mechanisms involved in nucleation, or the formation of the crystals, and the crystallization process itself. Development of experimental methods and predictive models which enable the production of improved crystals of biological macromolecules is of general interest. Improved crystals formation may ultimately lead to better drug development. In order to do the test in space, there's going to be several different types of hardware or containers that will be developed in order to run the test. The first one is ACE-M. ACE-M is a simple sample well container that will house the samples for either the colloidal systems or the biological systems. The second one will add a heater, where you can bring up the temperature inside the module to a specified level and then bring the temperature back down. In the third system, we're going to have very specific and unique temperature control allowing controlled temperature gradients up and down within a reasonable level. In the fourth system, we're going to have an electric field. In this system, you'll be able to impose electric field using these specialty cells and study the phenomena of the colloidal system or the macromolecule in that manner. And in the fifth one, which is going to be of most interest to most scientists, is the confocal microscopy. This system is currently under development and won't be ready until 2016. In confocal microscopy, there's going to be a confocal head, a confocal camera, a 532 nanometer double YAG laser, which will become available to provide 3D imaging of the samples. For example, fluorescently dyed core-shelled particles. With the advent of the LMM and the enhanced capabilities currently under development, we hope that research such as yours will yield new discoveries in colloidal systems or biological macromolecules. The forthcoming NRA provides a great opportunity to conduct research on the ISS. You can respond directly to the NRA or contact me if you have any questions. We'll put you on the right path.